Hello, BIOM 2131 students. This is Dr. Crozier, and I plan to run through the 45 questions of Exam 2. I'm going to move through these fairly quickly. I will pause for a couple of the questions that, on average, students had more trouble with. But for most of these questions, the class average was over 90%, so I don't feel that I need to dwell on other options too much, but I want to make sure the questions are clear. Also, you can look at the timeline at the bottom of this video, and you can see I've broken it up into segments so that if you want to scroll ahead and look at a group of questions that are later in the exam, you should be able to find that section on the timeline. So let's move through this quickly. The order of your questions may be slightly different depending on which version you had, but the content of all 45 questions is the same for all students. So first, I do want to point out on the exam for Exam 3, the same has been true for Exam 2 and 1, that I give each question a title, and the abbreviations like MC is just for multiple choice, and then the topic that the question relates to is here, if that helps you at all in completing the questions. So I do have drag and drop, so it does come out as D and D. There's also drop down, which I'll just call DD, but MC is for multiple choice. And so these just refer to the format of the question, and this is a question about mRNA stability. So you can look for that going forward on Exam 3. But which of the following statements about mRNA half-life is false? It is false that the half-life of most eukaryotic cells, mRNA, is greater than 24 hours. Typically, they're anywhere from a few minutes to 10 hours or so. Question number 2, multiple choice about protein coding genes. The human genome encodes 21,000 protein coding genes. Approximately how many of such genes does a typical differentiated human cell express at any one time? Straight from the textbook, most cells express between 5,000 and 1,500 or 15,000 of these genes at any given time. Some of these genes are never switched on, so it can't be all of them, and not even 90% of them. But at any given time, anywhere between 5,000 to 15,000 genes are being expressed. What differentiates one cell type from another is the particular genes that are being expressed, but there are a lot of housekeeping genes that are expressed by all cell types. Question 3, multiple choice about post-transcriptional control. Which of the following is not involved in post-transcriptional control? Well, all of these are, that is, they affect mRNA, except mediator. Mediator is a protein that helps in initiating transcription. Question number 4, multiple choice about RNA function. MicroRNAs, tRNAs, and rRNAs are all fill in the blank, so it has to apply for all of them. These other answers do apply for one or some of them, but the only one that applies for all of them is that they do not code for proteins. That would be true of mRNA. Number 5, multiple choice about the TRP operator. The tryptophan operator binds to the tryptophan repressor when the repressor is bound to tryptophan. That is, it's an allosteric protein and binds the repressor when tryptophan is present. We don't want to turn on the genes for making tryptophan when you have tryptophan already present. Number 6, this is a question that stumped a number of people. Ungated ion leak channels are always open. We often think of leak channels as being like open doors with no door on them, as if things can just flow through. The reality is they are not always open, and it's a misnomer. And so I spent significant time on this when talking about the patch clamp experiment. So I'm just going to jump over and play a video here for you. And this is from me teaching on March 9th, where I talk about this. I'm just going to play 1 or 2 minutes of this to answer this, because I suspect a lot of students wonder why their answer was incorrect. They chose true, but it's not true. They randomly snap between open and closed states, even leak channels.
So something that a lot of people don't know about these channels that I mentioned already is that these channels are proteins that actually fluctuate in their shape randomly. And they snap between open and closed states. And there's a number of experiments that have demonstrated this. And this is called the patch clamp experiment, where literally you take a glass tube and a vacuum, just zoom in on this, and this is a pipette on the end of a vacuum. And you can bind it to a piece of membrane and suck away a piece of the membrane and pull off a piece of membrane. You'll capture some channels in it. And then you can put that in a beaker with a known charged number of ions, and you can measure the voltage in the volume above the membrane and in the volume below the membrane in that beaker. And you can actually see that there's changes in voltage And that's because ions can flow through this when it's open and they can't when it's closed. And sometimes there'll be no movement, and then there'll be movement, then there'll be no movement, then there'll be movement. And that's because this channel is just kind of fluctuating in open and closed states randomly. And so this is what this experiment describes. I think there's a video on this, but they have a recording microelectrode. The suction pipette here is suctioned to a piece of membrane. They're measuring the voltage inside. When you pull that off and you put it in a beaker, so here we have that vacuum. We've pulled off a piece of the membrane, and it has a channel or multiple channels in it. And we're measuring the voltage inside in the solution here. And what we find is the voltage changes, it fluctuates. It goes up, it goes down. It goes up, it goes down. It goes up, it goes down. And the only way that can be is that under certain conditions, ions are flowing in or they are not. And if you look at this graphic up close, you can see what's happening is when we're measuring the current, we're running an electrode through this. When there's no flow, it's because the channels are closed. And when we get this spike and a change in the current, it's because the channels open. And it's closed, and then it's open, and closed, and open, which tells us that this is opening randomly, these channels. All right. So why do they open and close? Well, some of them just do this spontaneously, like leak channels. Leak channels just kind of open and close randomly, and don't need any type of stimulus to do so. But different types of stimuli can influence the opening and closing of ion channels.
Okay, so there you have it, where I explained that in class. And that's what I was asking about. This was a bit of a tricky question, I suppose, but it's just something that was clearly explained to you in class. And that's why it's not true that they're always open. It's false. All right, so if you assume that this is just a misnomer, it's something a lot of people think about leak channels, it's not true. Of course, voltage-gated and chemically gated and stress-gated channels do require stimulus to open, or tend to be more open under certain stimuli conditions. But even leak channels randomly fluctuate between open and closed states. So if you got that wrong, that is why. Number 7, multiple choice about microRNAs. Which of the following statements about microRNAs is false? MicroRNAs are produced from ribosomal RNAs. That is a false statement. They are transcribed from genomic DNA, just like any other gene. They are made by RNA polymerase, just like any other gene. And one microRNA can regulate the expression of many genes so long as it's complementary to those mRNAs. Question 8. This is a drag and drop question, fill in the blank, about membrane transport. A molecule moves down its concentration gradient by passive transport, but requires active transport to move up its concentration gradient. Transporter proteins and ion channel function in membrane transport by providing a hydrophilic pathway through the membrane for specific polar solutes or inorganic ions. Transporter proteins are highly selective in the solutes they transport, binding the solute to a specific site, changing conformation so as to transport the solute across the membrane. On the other hand, ion channels discriminate between solutes mainly on the basis of size and electrical charge. Question 9. Indicate whether the following statements are true or false. Although cholesterol is a hydrophobic molecule, it has a hydrophilic head group like all other membrane lipids. That is true. Phosphatidylserine is the most abundant of the phospholipids found in the cell membranes. That's false. It would be phosphatidylcholine. Glycolipids lack the glycerol component in phospholipids. That is true. And the highly ordered structure of the lipid bilayer makes its generation and maintenance unfavorable. That is false. It makes it energetically favorable. Question 10. Again, a drop-down ion channel question. The acetylcholine receptor in skeletal muscles is a ligand-gated ion channel, the ligand being acetylcholine. Stress-gated ion channels are found in the hair cells of mammalian cochlea. That's in the ear. Voltage-gated ion channels respond to changes in membrane potential. And many receptors for neurotransmitters, like acetylcholine, again, are ligand-gated channels. Question 11. Multiple choice about calcium pumps. Calcium pumps in the plasma membrane and endoplasmic reticulum are important for preventing calcium from altering the activity of molecules in the cytosol. When calcium is present in the cytosol, it binds to proteins that often are activated or controlled or regulated by calcium. And so by pumping it out of the plasma through the plasma membrane or pumping it into the endoplasmic reticulum, it prevents calcium from activating or changing the activity of those proteins in the cytosol. Question 12. Active transport requires the input of energy into a system so as to move solids against their electrochemical and concentration gradients. Which of the following is not one of the common ways to perform active transport? Well, we saw light-driven examples with bacteriorhodopsin. We saw sodium-coupled, like the sodium-coupled sodium glucose transporter. We see ATP-driven ones, like the sodium-potassium pump, but we didn't see any examples where potassium was used to provide the energy to drive a pump. Question 13. Patch clamp, again, shows us that all of these things are true except the patch clamp method does not show us that single channel patch clamp recordings have demonstrated that gated membrane channels will only open and close in response to specific stimuli. We see that they do this randomly, right? And that's what the experiment has shown us, that these channels can open randomly. MicroRNAs are produced from precursor mRNA transcript. That's correct. So again, that's just a repeat of an earlier question. They have their own genes. Question 15, true or false? All of these are false except for the third answer. Let me quickly explain why. A protein can be embedded on the cytosolic side of the membrane bilayer by employing a hydrophobic alpha helix. Well, that's probably the toughest statement of all four of these options. And it is a little bit tricky. The incorrect term is hydrophobic. It's not hydrophobic, it's amphipathic. So if you got that incorrect, you could still get 3 of the 4 correct and get 0.75 out of 1, even if you got that incorrect. But the incorrect term there is hydrophobic. It should be amphipathic. A membrane protein that pumps ions in and out of the cell is classified as an enzyme. That's false. That would be a transporter or a pump. A protein that relies on protein-protein interactions to stabilize its membrane association is classified as a peripheral membrane protein because it can be dissociated without the use of detergents. That's true. The peripheral ones is where you have a protein bound to a membrane protein. And so the protein that's bound to it is peripheral. It's not integral. Finally, lipid-linked proteins are classified as peripheral membrane proteins because the polypeptide chain does not pass through the bilayer. That's false. It still has a component that's lipid, so they are lipid-linked, so they're still considered integral proteins, even though they don't go all the way through the membrane. The ones that go all the way through the membrane are transmembrane, but even lipid-linked proteins are still considered integral proteins as opposed to peripheral. Question 16. Again, drag and drop the correct answer into the blanks. For each of the following sentences, fill in the blanks with the word or phrase that best selected from the list below. Not all words or phrases will be used. Each word or phrase should be used only once. There are several ways that membrane proteins can associate with the cell membrane. Membrane proteins that extend through the bilayer, again, are called transmembrane. Transmembrane proteins and have hydrophobic regions that are exposed to the interior of the bilayer. On the other hand, membrane-associated proteins do not span the bilayer and instead associate with the membrane through an alpha helix that is amphipathic. So there's the answer to the previous question. If you weren't sure if it's true or false, here's the answer. Other proteins are covalently attached to lipid molecules that are inserted in the membrane. Peripheral membrane proteins are linked to the membrane through non-covalent interactions with other membrane-bound proteins. Question 17. Multiple choice about bacteriorhodopsin. Explains bacteriorhodopsin protein. Which statement best describes how bacteriorhodopsin operates? The absorption of sunlight, there's your energy source, triggers a shift in the conformation of the protein's 7 membrane-spanning alpha helices, which is correct, allowing a proton to leave the cell. Question 18. Plasma membranes are extremely thin and fragile, requiring an extensive support network of fibrous proteins. The network is called the cortex. The specific protein that we talked about binding to this in red blood cells was spectrum, but that'll come up later. Question 19. About RNA interference. Which of the following statements that RNAi is true? RNAi is induced when double-stranded foreign RNA is present in the cell. So it's sort of a defense mechanism to deal with pathogens or deal with viruses used by cells. Question 20. Multiple choice about sodium ion channels. If sodium ion channels are open in a cell that was previously at rest, how will the resting membrane potential be affected? Well, sodium is in high concentration outside the cell, so it would flow into the cell. Sodium is a positive ion, so the cell resting membrane potential will become more positive. That is, it would depolarize. Question 21. But red blood cell membranes, here's that question. Red blood cells have been very useful in the study of membranes and protein components that provide structural support. Which of the following proteins is the principal fibrous protein in the cortex of red blood cells? So if you didn't know the answer cortex before, that is the answer to the earlier question. And specifically, the fibrous protein that's the principal one used here is spectrin. Question 22. Consider the apical location of a particular protein. So here it is, protein A in image A, expressed in epithelial cells, illustrated in figure A on the left. Which type of defect described below is most likely to cause a redistribution of that protein around the entire cell? Here we have protein A all around the apical, the lateral, and the basal membranes, shown here in figure B on the right. Well, what's keeping protein A on the apical surface only is this diffusion barrier, which are the tight junctions. So how could we arrive at situation B? Well, if we had deletion of the tight junction, then these proteins could drift in the membrane to the lateral and basal surface. Question 23. Drag and drop concerning prokaryotic genes. The genes of bacterial operon are transcribed. So the genes are transcribed into a single RNA. It's polycistronic. Many bacterial promoters contain a region known as an operator, right? So that's within the promoter, to which a specific transcription regulator binds, or it might be just upstream of the promoter. Genes in which transcription is prevented are said to be repressed. Interaction of small molecules such as tryptophan with allosteric DNA binding proteins, again answered earlier in the exam, such as a tryptophan repressor, regulates bacterial genes. Genes that are being constitutively expressed are being transcribed all the time. It means they're always being transcribed. We use the word constitutive for that. Again, this is a question I want to draw your attention to. It's meant to be worth 1 point, and each of these answers worth 0.2 out of 1. So if you get them all correct, you get 1 out of 1. The software gave them each a value of 1 point. So even though there's 45 questions, at this point, because it's worth 45 or 5 points, that would be 49 points. This happened again in a later question where instead of being worth 1 point, it's worth 2. But the class average on this question was still something like 94%. So your grade might show as a grade out of 50. This is partly why. And I just converted it to a score out of 15. I just took your percent on the exam, multiplied it by 15 to get your grade. I apologize for that. I'll try to catch that for exam 3. Question 24. True or false? All of these are false except one. Epithelial cells membranes are asymmetric. Proteins from the apical side of the cell membrane cannot diffuse into the basal side of the membrane. That is true. The rest of these are false. When a mouse cell is fused with a human cell, the movement of respective membrane proteins is restricted to the original locations at the time of fusion. That's false. They drift throughout the membrane surface. The only role of carbohydrate layer on the cell surface is to absorb water, which creates a slimy surface and prevents cells from sticking together. That's not the only role of the carbohydrate layer. That may be true, but it's not the only role. They can act as identity markers, for example. The longest carbohydrate chain found on the surface of cells are linked to lipid molecules. No, we see these glycoproteins and proteoglycans especially that have really long carbohydrate chains. Question 25. Membrane fluidity. Membrane fluidity is going to increase the shorter the fatty acid chain and the more points of unsaturation. So a shortest chain is going to be 15, and then with 2 points of unsaturation, it's going to be polyunsaturated. This is going to be the most fluid membrane. The longer the chain and the fewer the points of unsaturation, the more rigid or stable or less fluid the membrane will be. Right, so the most highly mobile phospholipid has a short fatty acid chain and multiple points of unsaturation. Extracellular fluid. What is a principal anion in the extracellular fluid that offsets sodium ions? It's chloride ions. Chloride ions are in high concentration outside of cells. Again, membrane fluidity. The lateral movement of transmembrane proteins can be restricted by several different mechanisms. Which mechanism best describes a process by which nutrients are taken up at the apical surface of the epithelial cells that line the gut, released from the basal and lateral surfaces? We have proteins that import glucose into the cell on their apical surface, which are not found on the basal or lateral surfaces. And so those proteins' movement is limited by the presence of a diffusion barrier, as depicted in the image several questions earlier. The lac operon, question 28. Which of the following statements about the lac operon is false? Lac repressor binds when lactose is present in the cell. That is false. When lactose is present in the cell, it binds the repressor, removing it from the promoter so that we can transcribe the lac operon, which produces enzymes that, among other things, can break down lactose. Question 29. Negatively charged ions are required to balance the net positive charge from metal ions such as potassium, sodium, calcium. Which of the following negatively charged ions is most abundant outside the cell, and which ion does it most often neutralize? So again, this question was already asked. Chloride ions are the most negative outside the cell, and they offset, in particular, not calcium, but sodium ions. All right, so they create a state of electrical equilibrium by balancing the positive cations, namely sodium. Question 30. MicroRNAs, the extent of complementarity of a microRNA with its target mRNA determines whether the mRNA will be immediately degraded or whether the mRNA will first be transported elsewhere in the cell before degradation. If there's a really high fidelity match, it's most likely going to have an extensive binding and double-stranded region and degrade it quickly. If it only has minimal match, typically it takes time before the mRNA actually gets degraded, and so it's first transported elsewhere before degradation. 31. Nucleosomes. Which of the following statements about nucleosomes is true? Histone acetyltransferases affect transcription by both altering chromatin structure to allow accessibility to the DNA and by adding acetyl groups to histones that combine proteins that promote transcription. This is true. (32) Drag and drop membrane lipids. The special function of different membranes are largely determined by the proteins they contain. Membrane lipids are amphipathic molecules composed of hydrophilic portion and hydrophobic portion. All cell membranes have the same lipid bilayer structure, with fatty acid tails of the phospholipids facing into the interior of the membrane, and the hydrophilic heads are on the outside. The most common lipids in most cell membranes are phospholipids. The head group of a glycolipid is composed of sugars. Again, each of these is worth just a fraction of a point. So if you got one wrong, you could still get multiple points, but you may not get a full 1 out of 1 if you got any of these incorrect. Question 33, diffusion through lipids. Which molecule will be able to diffuse through a lipid bilayer more easily? You might expect, because of the small charge, a hydrogen ion would flow more readily through a phospholipid bilayer, but here we have a larger molecule, but it's not charged, and it will actually diffuse through a membrane more readily than a charged ion like a hydrogen proton. Question 34. The distinct characteristics of different cell types in multicellular organisms mainly result from the differential regulation of gene expression, as transcription of genes that are transcribed. Of course, these are protein coding genes transcribed by RNA polymerase II. 35. Here we have a list of molecules of different chemical properties. Knowing that all molecules eventually diffuse across phospholipid bilayer, sort the list by dragging each item to the correct order that most accurately predicts the relative rates of diffusion of these molecules, with the fastest at the top, the slowest at the bottom, and in the correct order. And so here we have a steroid, so it's lipid soluble. That will move through more quickly than the others. We have a non-polar molecule like propanol. We have an amino acid, and then we have a charged ion. Right? So the fastest would be the steroid, the slowest would be the charged ion like sodium or potassium or any of these hydrogen ions that we saw in the earlier question. Gated channels. Which of the following best describes the behavior of a gated channel? It opens more frequently in response to a given stimulus. Yes, voltage gated, voltage will be the stimulus. Mechanically gated stress or physical mechanical stresses will be the gate. Ligand gated or chemically gated, that will control it. Regardless of what type of gated channel, it more opens more frequently in response to a specific stimulus. Again, drag and drop the correct answer from these options. MicroRNAs are non-coding RNAs that are incorporated into a protein complex called RISC, which searches the mRNA in the cytoplasm for sequence complementarity to that of the microRNA. When such a molecule is found, it is then targeted for destruction. RNAi is triggered by the presence of foreign double-stranded RNA, which are digested by the dicer enzyme into shorter fragments, approximately 23 nucleotide pairs in length. Question 38, multiple choice about ribosomal binding sites. Using genetic engineering techniques, you remove the sequences that code for the ribosomal binding sequences of the bacterial LAC-Z gene. The removal of these sequences will lead to translational inhibition. If we don't have the ribosomal binding sequence in the mRNA, then you can't translate the mRNA. 38. Some cells have aquaporin channels to facilitate the flow of water molecules through the plasma membrane. For these cells, what regulates the rate and diffusion of water diffusion across the membrane? Solute concentration. Water flows toward areas of higher solute concentration. 40. A hungry yeast cell lands in a vat of grape juice and begins to feast on the sugars that are producing carbon dioxide and ethanol. In the process, there is a chemical reaction. Unfortunately, the grape juice is contaminated with proteases that attack some of the transport proteins. So these are enzymes that break down the transport proteins in the cell membrane, and the yeast cell dies. Which of the following could account for the yeast cell's demise? Well, if it doesn't have transport proteins, then it can't move sugar into the cell, so it doesn't have nutrients to break down to produce ATP. Here's a question about the central dogma. Drag and drop these options into the correct slots. So DNA to RNA, that's transcriptional control. RNA to mRNA, so pre-mRNA to mature mRNA, that's RNA processing control. mRNA translated into protein, that's translational control. Once you have a protein, you can control whether it's active or inactive, that's protein activity control. Question 42. True or false? They're all false except for one. Most ion channels are gated, which allows them to open and close in response to specific stimulus, rather than allowing the constant unregulated flow of ions. That's true of gated channels. The rest are false. Aquaporin channels are found in the plasma membrane and allow rapid passage of water molecules and small ions in and out of the cell. Note the ions are selectively removed and only the water molecule goes through. Again, you can watch the video on aquaporins that I posted and I played in class. Gap junctions are large pores that connect the cytosol to the extracellular space. No, they connect the cytosol of one cell to the cytosol of another cell. The ion selectivity of a channel depends solely on the charge of amino acids lining the pore inside the channel. That's false. Also, the size of the ion is important. Question 43. Gated channel types. The stimulation of auditory nerves depends on the opening and closing of channels in the auditory hair cells. Which type of gating mechanism do these use? It's called mechanically gated, or as I said in class, mechanically gated is also called stress gated. So it's not ligand, it's not voltage. We didn't distinguish intracellular and extracellular ligand, but both of these are ligand gated. It is a mechanically or stress gated channel. Question 44. Please note that this has 2 blanks, and you're to drop the correct answer in, and it was worth 2 points instead of 1. That's just an accidental feature, and this is why your exam is now worth 50 points, even though there's only 45 questions. So, new membrane phospholipids are synthesized by enzymes that are found on the cytosolic side of endoplasmic reticulum. That's just a straight sentence from the textbook. If you got one of these wrong, you get 1 out of 2 for this question. The final question, question 45, fill in the blank, drag and drop to fill in the blank. For an uncharged molecule, the direction of passive transport across the membrane is determined solely by its concentration gradient. The electrical gradient is irrelevant because it's uncharged. On the other hand, for a charged molecule, the membrane potential must also be considered, the electrical component. The net driving force for a charged molecule across the membrane therefore has 2 components. It's referred to as the electrochemical gradient. Active transport allows the movement of solutes against this gradient. The transporter proteins called coupled transporters use the movement of one solute down its concentration gradient to provide energy to drive the uphill transport of a second solute. When this transporter moves both ions in the same direction across the membrane, it's considered a symport. If the ions move in opposite directions, the transport is considered antiport. That is all 45 questions. Again, your grade was initially out of 50. I simply took your percentage, say 92%, and multiplied it by 15 to give you a grade out of 15, which shows in your Blackboard grades. If you have any questions about any of these specific question content, feel free to raise it at the beginning of our next class or subsequent classes. I'll do my best to answer your questions at that time.